think I've made reference, at least on this channel, I know I have on the main channel as well, that using mouse studies, okay, lab rat studies, lab mouse studies, okay, can properly, most of the time, mimic the results that you would find in human beings. It's one of those weird crossovers that you get from lab experiments on animals that transfer to human beings. It's weird, right? But it's far more ethical to be experimenting on rats than on people. So what we can learn from some of these stuffs and what we can learn specifically about mouse models and autism should be important because, okay, listen, I'm not one of those original anti-vaxxers. You know the ones that we're talking about, the Jenny McCarthy's of the world, where any needle results in autism in my kid. That's retarded, and I wouldn't ever dream of promulgating that idea, mostly because I think it's ridiculous. But I'm not walking back my stance on that at all, so don't worry about it. But you can't deny that there's been a rise in autism diagnosis in children. Okay, is that from faulty diagnostic tools? Perhaps. But is it something that we can get to the bottom of by better understanding autism? Yes, of course. So if we can use mouse studies to figure this out, why not? Autism is a complex neurodevelopmental disorder where the incidence have been rising dramatically in the past 2 decades, in step with a dramatic rise in the use of glyphosates, the active ingredients in pervasive herbicide Roundup on core food crops. While correlation does not necessarily mean causation, there are multiple mechanisms by which glyphosates disruption of human biology and the biology of the gut microbiome. That's what's very important, and that breakthrough about a decade ago was incredible. Could cause many of the observed symptoms and biological metrics associated with autism. Remarkably, mice can acquire a syndrome that looks a great deal like human autism. Ah, cool. They expertly solve Rubik's Cubes. That's sweet. And researchers can be able to create multiple breeds of designer mice that exhibit autism like socio-communicative deficits. These mice strains have turned out to be very useful in helping us understand the pathology of human autism, even though the mapping is not perfect. One such strain is a naturally occurring inbred strain known as BTBRT plus TF/J mice. BTBR for short. Thank you for having a slight abbreviation for that. Anyways, so glyphosates. Why are they important to this study? Well, glyphosate is used extensively in agriculture, both in genetically engineered Roundup ready crops and of other core crops such as wheat and sugarcane. Why do I always advocate for less sugar and less gluten in your diet? Well, mostly because, well, they're just bad for you anyways. And then also for the unintended consequence of also consuming glyphosates. Not saying that you'd acquire autism, but if you want to go ahead and you want to try to eliminate that from happening, be my guest. Our food supply is highly contaminated with glyphosates, and so many children in America, sorry, not the United States, America, are exposed daily to this toxic chemical. The latest number coming from the Center for Disease Control on Autism rates in the United States is that 1 out of every 44 children as of 2021 is autistic. That's ridiculous, higher than any previous years. Heparin, sulfate, and the brain ventricles. The fact that a manipulation so specific to heparin sulfate in the brain is enough to induce autism in mice suggests that brain deficiencies in heparin sulfate may be a key central pathogen or pathology in human autism. Indeed, many genetic mutations linked to autism involve enzymes associated with synthesis of so-called extracellular matrix. This is a non-cellular component of tissues and organs, which not only provide physical scaffolding, but also initiates the orchestration many biomechanical and biochemical cues that govern cell physiological responses to environmental stimulants. A number of mutations linked to human autism occur in the set of genes that are called glycogenes, which encode the proteins and lipids that are bound to heparin sulfate in the matrix, forming heparin sulfate protoglycanes, HSPGS, interesting, or enzymes involved in glycosylation, the binding of heparin sulfate and similar complex sugar chain molecules to the proteins and lipids. I won't bore you with the rest of that stuff, but that's what they found in mice. So BTBR mice. What about gut issues? Interesting. Seminal study, all right, of the Bilber mice revealed specific disruptions in the gut that were hypothesized and led to neurological effects through interactions along the gut-brain axis. Normally, the liver synthesizes bile acids from cholesterol that conjugates them with either taurine or glycine before shipping them out of the gut or buffering them in the gallbladder. If it is the responsibility of the specific species of the gut bacteria, mainly Bifidobacteria, to deconjugate the conjugated bile acids, freeing up the taurine or glycine molecule for further metabolism. I'll skip ahead a little bit. The Bilber mice were found to have a deficiency in bile acid synthesis in the liver, as well as further a deficiency in the deconjugation and their conversion to a secondary bile acid in the microbiota. Very interesting. Did glyphosates cause autism in the BT-BR mice? It's easy to argue that these abnormalities could be caused and due to the fact of glyphosate exposure in the mice. The mice are progeny of multiple generations of inbred lab mice that were almost certainly fed a steady diet of glyphosate, and the mice feed manufactured are genetically modified Roundup Ready corn and soy crops. Never eat soy, not even once. A reduced supply of bile acids in each generation and direct toxicity of the city of glyphosate to certain species of bacteria would alter the microbial distribution over time. Thus, the gut microbiomes that were passed on from generation to generation could maintain a pathological distribution influenced by glyphosate acting as an antibiotic and enzyme disruptor. So did it? BT-BR mice also exhibited impaired serotonin synthesis, which resulted in slowed peristalsis and issues of constipation and small intestinal bacterial overgrowth. Furthermore, BT-BR mice had reduced levels of acetate in the gut, a short-chain fatty acid normally produced by gut microbes, especially Bifidobacteria during fat digestion, and an important fuel that feeds the Krebs cycle to produce energy. Acetate deficiency in the gut has also been seen in human autism. So, studies on mice exposed to glyphosate. This was exposure of male mice to glyphosate-based herbicides during juvenile and adult periods led to a marked reduction in serotonin levels and several nuclei in the brainstem. Argued in the paper published in 2015, the glyphosate could collaborate with aluminum to induce both gut dysbiosis and disruption of the pineal gland function in the brain. The pineal gland produces sulfated melatonin and distributes it into the cerebrospinal fluid of the ventricles during sleep. We propose that an important role for melatonin is to deliver sulfate neurons to boost that sulfate supplies to the HSPGS. Heparin sulfate plays a significant role in the clearance of cellular debris, which is an important aspect of sleep. And sleep disturbance is a common feature of autism. So this is getting close to closing the gap between heparin sulfate deficiency observed in the brains of BT-BR mice and their gastrointestinal disturbances. So what about said taurine? Anyways, could it help? Could supplementing with taurine help? Curiously, human cells are unable to metabolize taurine. But dietary taurine can be converted into sulfate by gut microbiomes. The brain, heart, and liver all store large amounts of taurine. Taurine released into circulation during encephalopathy, brain swelling, or during a heart attack. Taurine taken up during the liver and conjugated to bile acids. Glyphosate damaging effects on bifidobacteria would interfere with the production of sulfate from taurine by gut microbes, due to impairment in the ability to detach taurine from bile acids. So long and short of it, I was tired of dragging everybody through the weeds when it comes to these scientific studies. A recent study by William Shaw involved a set of triplets, 2 boys and a girl. Both boys were diagnosed with Tism, and the girl had a seizure disorder. All 3 children were found to have high levels of glyphosate in their urine. They also overrepresented of Clostridia species in the gut, which were suggested to contribute to the disease process through their release of toxic phenolic metabolites. Another study from 2017 in the gut microbiome of autistic children with inflammatory bowel disease compared to normal controls showed a reduced Blauta species impaired bile acid metabolism and increased in several species of Clostridia that were linked to reduced tryptophan levels and impaired serotonin homeostasis, along with overexpression of Th17, all consistent with various mouse model studies. So, it looks like there's some crossover once again between those mouse studies and human studies. Interesting. So, in summation, a disrupted gut microbiome, which could be caused by the glyphosate, leads to a leaky gut barrier, and the leaky brain barrier, and leaky placental barrier. This allows toxic substances such as aluminum, phenolic compounds, and glyphosate, as well as live viruses and endotoxins from vaccines, to invade the brain, and by breaching the placental barrier, exposes the fetus to harm. An overzealous immune reaction from these insults disrupts a neuronal development and causes autistic-like behavior in mouse pups and in children whose mothers have been similarly exposed. So, it's not the vaccines that are making your kids dumb or exceptionally good at pattern recognition. No, it's glyphosates. Very interesting. And I think of some of the major diseases that are in the world, I think we're probably closest to really getting a huge breakthrough. You see some of it when it comes to dementia and Alzheimer's. I wonder why the federal government is prioritizing that. It leaves a lot to be considered. But when it comes to one of the major diseases that are out there to really receive a monumental breakthrough, I think we're getting really close when it comes to the Tism. I'm hopeful for it, but not because I know anybody who suffers from it or anything like that. I just think, fewer diseases in the world is probably a good thing. But with all that said, thank you all very much for the gift of your time. I've been Don Consuelo. Why don't you follow your gut and get after it. Take care, everyone.